Good morning. My name is Amandrie Louw and I'm an MSc student at Stellenbosch University. So when you look at a natural landscape, you can see different plants and you can see different organisms. So plant production systems after the Green Revolution has modified these natural landscapes, by using monocultures over large areas, by applying chemicals such as fertilizers and pesticides, and then using conventional tillage practices such as a moldboard plow, and this all contributed to modifying soil structures. This all has contributed to the loss of biodiversity in agricultural systems and reducing natural services you would have gained from processes and functions from other organisms. However, with these external inputs, there's still an increase to crop vulnerability to pests and diseases, motivating the shift to a more sustainable agriculture. So a key strategy in more sustainable farming is then to reincorporate diversity into agricultural landscapes and to manage this diversity we have incorporated more effectively. Because by using this diversity, we can be able to maintain crop production, pest regulation, and soil health. The goal should be to encourage the use of agricultural practices that increases the abundance of functional diversity of organisms above ground and below ground that can contribute to these natural services we can benefit from. Conservation agriculture is a potentially viable system for sustainable intensification without contributing to more negative impacts on the environment and also not converting natural areas to agricultural areas. Conservation of no-tillage systems has been adopted by more than 70% of large-scale farmers in America, Latin America, and Australia. In South Africa, conservation agriculture is predominantly practiced in the Western Cape, as well as in KwaZulu-Natal, where other areas are relatively small in comparison to conventional tillage practices. Conservation agriculture is based on three principles. The first one is the use of minimum soil disturbance, mechanical soil disturbance, the use of maintaining a soil organic layer, and this is encouraged by the use of less soil disturbance. Then using crop diversification, either in sequence or in association. Conservation agriculture also complements other approaches of farming, such as livestock integration. This further diversifies crop rotation systems and acts as a risk management practice for farmers. Today, conservation agriculture is considered to deliver multiple natural or help maintain natural services. So during my MSc, I would like to investigate 2 out of the 4 broad categories, such as supporting and regulating natural services, where arthropods play an important role. Arthropods include insects, other invertebrate animals, and sometimes they are referred to as gogas. Some of the pictures which I will illustrate now are just a few of the organisms I already collected during my sampling process. You can divide these organisms into functional groups, which refers to where you group them on their lifestyle or their feeding habit. So this one, which I will illustrate now, plays an important role in decomposition processes in helping or making microstructures within the soil. Some of the other pictures which I will show now refers to natural predators that help regulate non-beneficial organisms. And then of course our non-beneficial organisms can also be grouped based on their feeding habit, such as sap-feeding organisms and leaf-feeding organisms. So my hope during my MSc is to get a better understanding of the non-beneficial organisms you find in field crops. Then I would like to understand how these different organisms will influence consecutively grown crops in crop rotation systems in South Africa. I would also like to understand how diversification in terms of livestock integration and crop species diversification within conservation agriculture will influence this beneficial and non-beneficial organisms. And then lastly, I would like to understand if there is a correlation between the agronomic performance, the arthropod dynamics above and below ground, crop health and climate. So during my MSc, data collection will take place, will be subdivided into 4 chapters. Chapter 1, I will look at the agronomic aspect, where I'm going to look at yield components such as plant populations, biomass, leaf area index, and seed quality. For my chapter 2, I will investigate above the beneficial and non-beneficial arthropods by using different trapping methods such as a sweep net. Sweep nets are generally used to give you it's an easy and economical and easy way to determine some of the organisms you find on plant material. Then I will also use pan traps that is used to attract flying insects that are color attracted. And then I will use pitfall traps that are used for soil dwelling organisms. Both pan traps and pitfall traps are filled with a trapping liquid. My last chapter will focus on remotely sensed data. This will give us an indication of crop health and give us just more information in terms of growth vigor from an outer perspective. In this picture you will see now, you will see the colors are indicated for low growth vigor and some colors are indicated as high growth vigor. The low growth vigor areas just gives us an indication that it has to be further investigated why performance is not at optimal stages. At the end of this MSc project, I would like to promote a system and management approach, such as diversification and crop rotations, as a tool to maintain crop production, pest regulation and soil health. Promoting these functional groups, which can contribute to ecosystem services or those natural services we can gain from their natural functions and processes. That the diversity of these invertebrates, animals, is an indicative of a broader sustainability in the whole cropping system. So in chapter 2 for above ground insects, I will use a sweep net, and this will just give me an indication about organisms you can find on the vegetation. Now this is a very simple and economical method used by entomologists. Another option for looking at above ground insects is then using pan traps. So pan traps are used for flying insects that are color attracted. So in my MSc I decided to use yellow, which is a very general color. Then the third option or the third method is using pitfall traps. Now this is mostly for soil dwelling organisms. So what you do is you place this level to the soil. You place it level to the soil, and then both these pan traps and pitfall traps are then filled with a trapping liquid. This could either be propylene glycol, which is a very safe liquid to use, and then also soap water. The soap in the water just breaks the surface tension, preventing the organism or insect to escape from the trap. The last method is taking soil samples to give us a representative of the beneficial and non-beneficial organisms in the soil. So basically you just take soil and then you throw it through this funnel with this sieve, and then underneath there is a little container with ethanol or a preservative liquid. And then slowly, as the soil dries up, the organisms within the soil will then fall through and then land within this little holder. So the main aim about my project is to really help farmers to get the bigger picture of what's really happening within the crop rotation systems and using diversification as a tool, just to promote natural services, but not influencing their yields.